Good morning, everyone. Let's get started. Okay, so in the past, we mainly focus on the single gene inheritance pattern, which means we focus on the simple traits. Now, today we're gonna introduce the inheritance pattern for the complex traits, or we can also call the quantitative traits. All right, what's the relationship between the simple traits and the complex traits? Also today, we're gonna introduce a little bit of basic statistic and statistical analysis that can be applied in genetic dissection. We're gonna also introduce genetic model and how we can use the genetic model to help us to calculate the heritability for any traits. And we're going to also introduce the quantitative traits mapping, loci mapping. So the complex traits, also called the quantitative traits. So how we can scan the entire genome to study a population and scan the whole genome to identify the location where the genes that are responsible for the traits is situated. So this is the topics for today. All right, first, let me introduce some basic concepts for statistics. Probably many of you have already taken some statistic classes, but if I ask you what is statistic, I don't know if you can answer this question or not. So today, don't worry if you cannot definitively answer this question. So let's study this concept today. All right, statistics or statistical method is a method that we can use to study a population. So suppose you are interested in finding out what is the average height of the human beings on all the people on this planet. So that's the population, the whole population. And you want to find out the average height of this population, right? Which is mu. So in this case, we say mu is the parameter of interest that we want to figure out for the population. As you can see, the population could be a very large, infinitely large. So you cannot afford this study, right? You cannot go to every single place to measure every single person and then get the number and then calculate average. So that's impossible for us. So what we can afford to do is to study a random sample. So a random sample means like we just take a random sample from the population of interest. This sample should be a random sample. Why? Because we need the sample. So we need a sample that can represent the population, right? So if you are not taking a random sample, if you took a biased sample, then the conclusion you made from the sample, from studying the sample, can be misleading. So may not be the best way to make a conclusion for the population. So okay, so first we need to take a sample from this random sample from this population. So this process is called the sampling or data collection. Once you have a sample, for example, you take a random sample from the population, you can simply calculate the sample mean, right? Sample mean, X bar, sample mean. And this X bar, this sample mean is called statistic, all right? Okay, so this is the definition for the statistic. So if you take a random sample and if you calculate sample, if you calculate a summary score for a sample, then that summary score of the sample is called the statistic. You can use this statistic, which is summarized from your sample, random sample, to infer the population parameter of interest. So this whole process is called the statistical analysis. Let me repeat. So we are interested in this population, and we want to calculate the parameter for this population. what we do is like we take a random sample from this population. Oops. And if you have a random sample, you can calculate the sample summary score, like sample mean, and use this to go back to the population to infer the population parameter. So this is the way we do in the statistical analysis to study a population of interest. All right. Okay, so let's go back to our business. This is the classical dihybrid cross, which was performed by Mr. Mendel, right? All right, this is a statistical analysis. Actually, in the history of genetics, a lot of famous geneticists, they apply statistical methods, statistical analysis to help them to develop, to find rules, principles, and develop and help them helped them to develop many famous laws, in genetics. So this is an example here. All right, let's look at this statistical analysis. So actually, Mr. Mendel is actually interested in the F2 population, right? So this is a classical dihybrid cross. Okay, now he's very interested in the F2 population. If these two genes, so this experiment was done to demonstrate if these two genes are assorted independently, right? If this is true, so these two genes should be located on two separate chromosomes. Okay, so this is what we want to test. All right, now if this is true, if the null hypothesis is true, so if the model is true, or if the hypothesis is true, so we expect to see this ratio, right? This ratio in the phenotypes. So for the entire F2 population, we expect to see this phenotypic ratios, 9:3 to 3:1, so famous ratios. However, the entire F2 population is too large, infinitely large. So we cannot sample every single seed from this cross. What we can do is like we can take a random sample. Of course, the size should be large. The sample, this is a random sample. And if we want to get a reasonable or accurate estimation, so we hope to get a large size random sample. So this is a random sample. Now you have your data, you have your sample, and you can calculate the phenotypic ratios in your sample, right? So this ratio will never be exactly the same with the expected ratio. But if the sample ratio is very close, it's close enough to the expected ratio, then you have strong evidence to say, okay, your null hypothesis is true. And this sample ratio is equivalent to the population ratio. And then we can say, we can make a conclusion. Yeah, we believe these two genes are sorted independently during the meiosis. All right, so this is a statistical analysis. As I mentioned here, so the expected ratio here is the parameters of the population, F2 population. We do not know these parameters, and we want to estimate this. Then we can take a sample, and we use the sample, the phenotypic ratio in the sample to estimate the expected ratio in the population. Right? Okay, so this is the way how we can perform a hypothesis testing to use your data to test the null hypothesis, right? All right, in this case, so here I'm gonna give you introduce another concept here. So distribution. All right. So in this specific dihybrid example, the distribution of the phenotypic categories is like this. Distribution is because we have 9:3 to 3:1 ratio. So we have 4 categories. So the distribution is like this, and this is a categorical distribution. We have 4 categories here. Right. So let's look into the relationship between the simple trace and the complex trace. All right. So as we studied before, so Mendel actually hundreds years ago, so Mendel actually did a lot of study. Okay, it's here. So he actually chose to analyze 24 traits, but finally he only reported his research based on 7. Why? Because those 7s, the data collected for those 7 traits are in agreement with his hypothesis. So yeah, but now we understand why only 7 traits can support his Mendelian laws, because those 7 traits are, all of the 7 traits are simple traits. When I say simple traits, means like that phenotype is only controlled by a single gene. We are very familiar with this, right? So either like if we look at here, so if we study the shape of the seeds, so either it's round or it's wrinkled. So, and this is simple, and this is only controlled by 1 gene. And if you look at the seed color, yellow versus green, that's also a simple trait, which is controlled by a single gene. And if this is a single trait, and if this trait is only controlled by a single gene, then it will give you a categorical phenotype, either yes, 0, either yes, 1, or no, 0. So we can easily put the sample into 2 categories. All right, the distribution of this phenotype will be like this, 3-to-1 ratio, right? This is the categorical traits. Now, how about the remaining like 18 traits? So what happened to those traits? Those traits, the data for those traits are not supporting his hypothesis. So he chose to not report those. But right now we know why is that, because those traits are not simple traits. Those traits are not controlled by a single gene. They are controlled by many genes, more than 1 genes. Okay, so that's why if you look into the other traits here, those traits can take many values, and those values, those phenotypic values, excuse me, if you look at those phenotypic values, they are behaving in a continuous way, the numerical way, like this. So if you plot the distribution of those traits, they will look like this. So this is very famous bell-shaped distribution, right? So probably many of you already knew the name for this distribution. This is a normal distribution and Gaussian distribution. Again, so the difference between these 7 traits and the remaining traits is the every single one out of these 7 traits is controlled by a single gene, but the remaining traits, they are controlled by many genes. So we call this complex traits as the quantitative phenotypes. All right, now let's look into this. What's the relationship? What is the relationship between the single traits and the complex traits? All right, let's start from here. So this is a simple trait, and this trait is only determined by a single gene. However, there's a new concept here. So incomplete dominance. So this is the first time we introduce this concept here. In the future, after the first midterm, which is next Tuesday, don't forget about that. That's important. Yeah. All right. So after the first midterm, so we're gonna use this concept a lot, because when we do the advanced genetic dissection, we're gonna talk about a lot of situations, cases involving the incomplete dominance. So what is incomplete dominance? It has another name called the co-dominance. dominance, codominance. Okay, which means, okay, so we are talking about one gene, right? Because this is a simple trait, and it is determined by only one gene. So for this gene, we have two alleles. Since these two alleles are following an incomplete dominance or codominance pattern, so what does that mean? That means like if we look at a heterozygous individual, which is the capital R and lowercase r, so in this individual, both traits will be expressed. So both traits will be present. So what does that mean? let's explain a little bit more here. So first, we can look at the homozygous, the big R. Okay, the big R give you the red color of the flower. That's one trait, right? And if we look at the homozygous little r, this will give you a flower without color. So the product from this gene will show no color, no pigment at all. So okay, now if we look at the heterozygous, so in this individual, both traits will be expressed. Means half of the product will from this allele gives you the red color, and half of the product from this allele in this flower will gives you this product. And if you mix this red stuff with the colorless stuff, this colorless stuff will dilute the redness of this. So this will gives you a pink phenotype here. So let me erase this. So this is different from what we have learned before, right? So in the past, we focus on the complete dominance. Complete dominance means if there's a big R, so the small r will be hidden. But this case, no. All right, so if this is a codominance case or incomplete dominance case, if you compare the phenotype of this and this, the homozygous big R or heterozygous, this will show you two different phenotypes. But this is different from what we have learned before, right? What we have learned before in the complete dominance situation, so this two individual will give you exactly the same identical phenotype. But now, in this case, which is the incomplete dominance, you're going to see two different phenotypes, right? The red and the pink, less red. I hope this is clear. All right, so this is a new concept here. So pay attention to this concept. What's the difference between this and the previous complete dominance genes, alleles? All right. So we're going to use this a lot after the first midterm. All right. Okay. Now, so in this case, so we can use Punnett square to help us to study the cross between these two parents, right? The homozygous, heterozygous parents. This is the mono-hybrid cross, and now the phenotypic ratio, the distribution of the phenotypic, the distribution of the phenotypic categories will look like this, right? 1, 2, 2, to 1. All right. Because we have very clear categories. So in here, in this situation, we only have 3 categories, right? 3 categories. So we can get this bar plot, the distribution. Okay, so this is one gene, simple traits. Okay, now let's extend this example to a little bit more complicated situation. So this time, so this is not a real example, but I just use this as a hypothetical example to show you the relationship between the simple traits and the complex traits. Okay, now in this case, the situation becomes a little bit more complicated. I cannot see this. All right. So suppose we have two genes. I cannot see the top of the slide. So let me, okay. All right, we have two genes. One gene is R1, little R1, which is on one chromosome. This is clear. We have another gene, which is called the R2, big R2. And the recessive, another allele is little R2. Okay, those two genes are located on two different chromosomes, which means they are assorted independently during the meiosis. However, those both genes are doing the same function in the cell. What does that mean? That means like this gene can produce the red particles, the red pigments, which can be deposited in the flower petal. And this gene can do the same thing, but they're doing the same thing independently, okay? All right, they are two different genes, but they are doing the same. They are doing the repeated function. All right, okay, now, then if we do the classical dihybrid cross, so we are gonna get this Punnett square here. So this is equivalent to the full, so we are very familiar with the 4x4 table, right? That Punnett square. Actually, this is the same with this 4x4. Why? Because still for each parent, we have 4 types of gametes, but this gametes will give you 2 doses of the red particle, the pigment, and these 2 gametes will give you 1 dose. Each of these 2 will give you 1 dose of the particle, right? This gives you a 0 dose. Okay, so we can group these 2 gametes to form 1 category here. So that's why for one parent, so this is one parent, the male parent. So it can produce like half of the gametes, which can produce 1 dose, and 1/4 of the gametes that can produce 2 doses, and another 1/4 produce nothing. All right, the same for this parent. So if you look into this Punnett square, and you can easily summarize like from among this F2 progeny, you have 1 category with 4 doses. You have another category with the 3 doses. You have number 3 category with two doses, and the number 4 with a single dose, and number 5 with 0 dose, with nothing. Okay, so if you summarize these categories, those will give you the distribution of the 5 categories. So see what happens here. So when we compare with the previous slides, with previous slides, we have 1 genes, right? Here, 1 genes, oops. Okay, now we have 3 categories. Is that right? So 1 gene, we have 3 categories. Distribution is 1 to 2 to 1. Now we have 2 genes. So how many categories do we have? We have 5 categories. We have more categories. Why? Why it becomes more complicated? Because we have more complicated situations, right? We have 2 genes. So now we have more categories. And if you look at the difference between 2 adjacent categories, the difference between any 2 adjacent categories become less. All right, this is the consequence. So is that clear? Still, this is a categorical phenotype situation. We can clearly divide the F2 progeny into 5 different categories based on their phenotypes. No doubt. Yeah, all right. Okay, so this is the 2-gene model. Now, if we extend this model into 3 genes, so 3 genes can do the same thing. What happened? So here we have more categories, even more categories. We have 7 categories. And if you look at the difference between any 2 adjacent categories, the difference between any adjacent categories in this case becomes even less. All right, imagine if you increase the number of the genes, so you're going to end up with many categories, like here. And if you look at the difference between any two adjacent categories, you probably cannot tell the difference between any two adjacent categories. That's why if a trait is controlled by many genes, you're going to end up with a continuous distribution, not the category, not the category distribution in the simple examples. So this is a relationship between a single trait and a complex traits or the quantitative traits. Yeah, the quantitative traits or complex traits means that trait is controlled by many genes. So from here to here. So I hope this is clear to everybody. So if very few genes are controlling a phenotype, it's easy to get a categorical distribution. However, if this situation becomes more and more complicated, we're going to end up with a continuous distribution, quantitative traits. Okay, so yeah, that's the take-home message. Quantitative traits. So today we are learning something different. In the past, so we start with, in the previous lectures, we studied from very simple model, simple traits, which is controlled by a single gene. But now, so we will migrate into a more complicated situation here, right? Quantitative traits or complex traits. So this traits is usually controlled by more than one gene. All right. So if in the exam, so I ask you what is the quantitative traits or complex traits? What's the difference between this and the simple traits? So I hope everyone can give me an excellent answer to this question. All right. So in reality, so in human genetics or other or genetic study for other organisms, usually we're dealing with complex traits. Because sometimes simple trait is too simple, so it's easy to figure it out. But the complex trait is very difficult. So those traits commonly are normally distributed. So the bell-shaped normal distribution. So if we want to study a specific distribution for a trait, we need to use a model. So the normal distribution itself is a model. Then when we try to use a model to describe a distribution, we need to estimate the parameters, right? Mu and sigma. Mu is the population mean, so we talk about this. Those are the population parameters. The mu and the sigma square is the variance of the distribution. So we need these two parameters to definitively describe a normal distribution. So in this example, as we can see, we have four different normal distributions in four different colors. Okay, so obviously the green one is quite different from the other three because the mu, the average, the mean of the green distribution is completely different from the other three, right? Yeah, so this one is significantly different from the other three. However, if we compare these three, the blue, the red, and the orange, so they are centered at the same location. They have the same mean, same average. However, they have different variance. So that's why these three are also three different, completely different distributions. So again, so when we try to describe a normal distribution, we need to use the center and the spread to describe this distribution, which means if we want to estimate a normal distribution, we need to measure the central tendons and the spread of this distribution. All right, so this is the way where we calculate the central tendons. It's easy, right? So once you get a sample from the population. So if you want to calculate the average height of dogs, and then, so you cannot measure every single dog on this planet. So the same, so we can take a random sample. And for the sample, we can calculate the sample mean. And this is called statistic. We use this sample mean statistic to estimate the population mean. Right? This is the parameter. This is the statistic. All right. So example here. So this is how, very simple. So I think most of you have already done this before to calculate the sample mean. And also we can use this formula to calculate the sample variance. And the sample variance can be used to estimate the population variance, which is the parameter. So the sample variance is actually called statistic. Okay. All right, normal distribution. So don't worry about these horrible equations. I will not test you on this. So this is only for the information. Okay, now, so analysis of variance. Okay, so this is a very useful tools. In the future, in the future, you're gonna so if you go to the graduate school, medical school, or pursuing some career in genetics or other fields, so you always you will always use this useful tool to help analyze your data. So analysis of variance, Anova. So what is this? Okay, so suppose we are interested in this population, and we want to the entire population, and we want to estimate the variance of the human heights. All right. So we can easily estimate the total variance, right? The variance of the heights, and this variance actually can be composed into two parts. So the variance we observed in the population, the variance in the human heights observed in this population actually came from two sources. So one source, of course, is from genes, right? Genetic factors. So different genes or different genetic background will give different people will give people different heights, right? And of course, even some people carry the same identical genetic background. For example, the identical twins, they may have different heights. So how can we explain that? So that difference actually is from the environmental area, right? So here I just want to show you that the total variance observed in the population can be decomposed into two parts. One is from the genetic sources. The other one is from the environmental factor. Okay, now, so this is the famous equation here. The total variance can be written as the sum of the variance due to the genetics and the variance due to the environments. Okay, now, so if you in the future, if you are taking other genetic classes, or if in the future you're gonna take some genetic related career, you're gonna use this term a lot, the heritability, H square. Heritability for any traits of interest. The broad sense heritability is defined in this way. So it's defined as a ratio. So in the numerator is actually the variance due to genetics. And the denominator is the sum of these two, right? That's the total variance. Okay, now if you look at the entire ratio, it's easy to define this term heritability. The broad sense heritability is equivalent to the proportion of variance that can be explained by genetics. So the heritability, the broad sense heritability is defined as the proportion of the total variance in the traits that can be explained by genetics only. right? So this is an important term. So pay attention to this, and it may show up in your next midterm, right? Okay, so let's apply these rules to some interesting problems here. So Yao Ming, I don't know if everyone knows Yao Ming. So he's very famous, and yeah, he's really tall. So today, so let's use this example to show how we can use genetic model to study the genetics in Yao family. All right. So look at here. So this is the distribution continuous quantitative trace, right? Which means human height is actually a complex trace. It is controlled by more than one genes. That's why you see the continuous distribution. So this is the distribution for all the women in this world. And this blue curve represents the distribution for men. And Yao Ming actually stands here. So really outstanding. Okay, so let's look into the genetics in Yao family. So Yao Ming actually came from Shanghai, China, Shanghai. So this is the distribution of the human heights. This is the distribution of the heights in Shanghai. In Shanghai men, all the men in Shanghai. All right, the average is about like 170 centimeter. Okay, so suppose Yao Ming's parents had many kids like Yao Ming. So and they're all tall, right? And if this was true, I think there would be another dream team in this world. All right, yeah, fantastic. Okay, so suppose this is the case, and his family has siblings like this, and so we can easily calculate the average of Yao family. All right, outstanding. So the 212 centimeter. Oops. All right. So now let's learn how we can use a genetic model to help us understand genetics behind this famous family, right? So this X is the phenotypic value for Yao Ming. Okay, this X equals to, cannot see the number because, yeah, the top slide was hidden. Okay, all right. So here, the X bar is the average man in Shanghai. So, okay. Of course, his phenotypic value will include the average man in Shanghai, and also plus a G. This is the contribution from Yao family, genetics of Yao family. So this part is the extra due to the Yao genetics, which is the extra contribution over the population mean. And this part is the environmental factor due to Yao Ming itself, because this will explain the difference between the Yao boys, right? Okay. So if we look at here, so what is the contribution from the genes of Yao family is actually the difference between the average of the family and the average of the entire population. Okay. So that's the contribution from the family. All right. Now, if you look at the individual, the Yao Ming's height is like this, but his family average is like this. So he is really lucky. So he got 17 centimeter extra due to the special environment. All right. So using this, so we can easily dissect what is the contribution from the gene. So this is only due to the gene, right? This is due to the environmental. So we are interested in this because we want to identify the genes that are responsible for this extra 42 centimeters when compared to the population mean. So this is what we can do to the genetic study. All right. Okay. So first we need to show, okay, the effects from the Yao family genetics is significant. So what we can do is like we can do a two-sample t-test. So probably some of you have already done this before, but not using this data, but using some other data to compare two groups. So here, if we want to show the genetic background of Yao family is really outstanding, so we can compare the Yao boys, the height of Yao boys with other Shanghai men. So the blue curve shows the distribution of other Shanghai men, and the orange curve shows the distribution of Yao boys from the Yao family. And of course, you're going to see a shift between these two distribution, right? The means of the two distribution are different, but we want to use the data to test whether this difference is real or not, is significant or not. So we can do hypothesis testing. So this is a different hypothesis testing. Now everybody is expert for in hypothesis testing, right? Yeah. So this is a thing we can do. All right. If we can construct a t-statistic, and when we look at the t-table, if we got a p-value equals to 0.0008, so what does that mean? That means like, yeah, if we assume there's no difference between these two distributions, if we assume the null hypothesis is true, the p-value is only this small, which means it's very unlikely, right? It's very unlikely that this is true. So if the p-value is this small, we're going to reject the null hypothesis. And this will determine, yeah, that so the difference between the Yao boys and the other Shanghai men is real, is significant. All right, so this is what we. Okay, now first we have shown that there's a significant difference between the Yao genetics, Yao genes and other genes. All right, so we need to find out what those genes are. So what genes in the human genome that can control the human height? So as we know, there are too many genes on human genomes, right? About like maybe 20,000 genes. So which one is which ones are responsible for human height? So what we can do is like we can do the quantitative traits loci mapping. So this is how we introduce this concept here. All right. So we can study a population. So any population, for example, like the Shanghai man, or including women as well. So, and this is the whole population. And for each individual in this in this sample, so we take a sample first from this population, and for each sample we can genotype. We can genotype the markers on the genome for each individual. So this is the markers. Okay, now so we can test each marker to see if the distribution of the marker is associated with the distribution of the phenotypes here. So let me give you an example. All right, so when we look at the first marker, so there are two genotypes. The red represent one genotype, let's say maybe homozygous AA, and the blue represent the other genotypes, heterozygous A prime. Now, these two genotypes will divide the entire sample into two subgroups, right? One subgroup is represented by red dots, and the other subgroup represented by the blue dots. Now we can compare the distribution of these two subgroups. So this is the one subgroup distribution, and this is another subgroup. If the distribution is well removed from each other, which means what? Which means, yeah, these genotypes can separate the sample into two subgroups with significantly different human heights, right? So this is the conclusion we can make. So if this is true, means, okay, so this gene is associated with the phenotype. And this is what we want to identify from the entire genome, right? However, for some other traits, let's say for some other markers, so here, let's say for these markers. So there are two genotypes as well. So either the homozygous CC or the heterozygous CC prime. And these two genotypes can also subdivide the same sample into two subgroups. But if you check the two distributions of these two subgroups, they are overlapping with each other, which means there's no difference between these two sub-distributions. What does that mean? That means like this genotype, the variation of this genotype is not associated with this phenotype. So this marker is irrelevant. Only this marker, the first one, is relevant to the phenotypic variation. So this is how we do the quantitative traits loci mapping in a population. Okay, so another example is like, so we can study tomato weight as well. So tomato weight is also a complex trait, and it could be controlled by more than one genes. And we do the same thing. We can get a random sample from the population, and to form a random sample, and we can test each marker to see if the genotypic variation for each marker is associated with phenotypic variation, tomato weights. So in this way, we can scan the whole genome to identify the genes that are responsible for the trait of interest. Okay, and after that, so imagine there are millions markers on the genome, so we can scan the whole genome, and at each locus we can calculate a test statistic like this, here, 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 and we can draw a curve to represent the entire genome. So this is the entire genome. At each locus we can get a test statistic, and we can draw this plot, and this is the threshold. So any statistic is higher than this threshold will be significant. So we can easily scan the genome and identify the region, genomic region that contain genes which are responsible for the trait of interest. Okay, so this is a tool, very useful tools, and in our lab, in my lab, we use this a lot. So GWAS, which is called the Genome-Wide Association Study. So as you can see, so this is a human example. So we have 22 autosomes and the X chromosome, right? So this is the entire genome, and there are too many markers. Probably there are about like more than 1 million markers on the genome. And for each marker, we can test its association with the phenotype. For example, disease with the disease, and make this plot. This is called the Manhattan plot. Why is that? Because it looks like Manhattan. It's beautiful and it's busy. So, all right. So we have a cutoff here. So any position shows the test statistic beyond this cutoff. This will give you an indication that this region on the genome is very likely to contain the genes that are responsible for this disease. And in the future, so this is only for information. In the future, we're gonna study how we can use the fine mapping by the recombination experiment to help us to identify the genes, the real genes that are responsible for the traits of interest, and also we can do a lot of experiments to validate those genes. All right, I think this is the last slide for today's lecture. So I'm gonna show you an example problem. This is so you can find this problem from the syllabus. So this is a solved problem in chapter 17, I guess. Okay, now in this problem we have two true inbred lines of the beings, and these two pure parents are intercrossed to create a F1 progeny. So now everyone knows this problem. So this cross to generate the F1 and so the monohybrid cross to generate F2, right? So we should be very familiar with this. All right. So the two parents, pure line parents, are crossed to generate the F1 population. So when we calculate the variance of the being weight, this variance equals to this. Okay, this is the variance for the F1 population. Now, next, when the F1 is solved or intercrossed, so to generate F2, the total variance in being weights for F2 progeny is this much, okay, 61. So here we want to estimate the broad heritability, broad sense heritability of the being weight in the F2 population of this experiment. So to answer this question, so we have to understand the behind concepts. So what is the broad sense heritability? Okay, so today we learned this, right? The broad sense heritability is defined as the proportion of the total variance observed, which can be explained by genetics only, right? That means like there's a ratio, proportion, right? So there's a ratio. In the numerator, so here, this is the variance that can be explained, by genetics. So it should be this, variance G squared. And the denominator should be the total variance. Total variance is the variance observed. And this can be written into this. The numerator is the same, but the denominator can be rewritten into the sum of these two. Okay. All right. So let's look back to this problem. The F1 progeny are of the same genotype. Do you agree with this? Because if we cross like the parent big R with the another parent small r, so to generate F1. So if you look into the each individual of the F1 progeny, they carry the same identical genetics, the big R, small r, heterozygous. So the genetic background for F1 progeny are identical. Then what can explain the variability observed in F1 progeny? Because they have the same genetics. The variance shown in F1 progeny population, F1 progeny should be only explained by environments. Do you agree with that? So this is the key point. So the problem gives you enough information, but you need to identify those information to help you answer the question. So F1 progeny have identical genetics. So any observed variability in F1 progeny cannot be explained by genetics difference because they are identical. The difference, the variability in phenotypes observed in F1 progeny is solely explained by the environmental factors, which means the variance E equals to 15. So that's the part of this, okay? This is 15. All right, now in F2, so think about F2. So for the F2 progeny, do they have identical genetics? No. So because if you do the monohybrid cross to generate a F2 population, there are different types of genetic makeup, right? When we look at the single gene, so in F2 we could have like big R, we could have big R, small r, and we can also have small r and small r. So the variance in the phenotypes, the phenotypic variation in the F2 progeny actually came from two sources. One is from genetic difference, and then the other one is also from the environmental factors, right? So, but the total, we observe the total is 61. So how can we get these two numbers here? All right. So if we assume the variance of the sigma square of the environments, the environmental variation is identical, is the same for F1 and F2 progeny, then this should be 15 as well. We have the equation like this, right? The VG plus VE equals to the V total. Then we have, if we have already known the V total and the VE, it's easy for us to get the number for the VG, right? The VG equals to 61 minus 15, which is 46. All right, this is the variance that can be explained by genetics. Then it's easy. So the answer is like the heritability, the broad sense heritability is defined as the ratio of this. This gives you the final answer. And the heritability for this trace is pretty high, 75%, which means if you see the variability of the population in this trace, if you see the variability in the trace in the population, so 75% of the variability can be responsible, can be explained, accounting can be explained by the genetics. Right? So this is how we understand this. So any questions? So for today, so after this, I'm gonna briefly review the problems in the practice test. So I believe everyone is interested in this part as well, right? Before we move on to this part, so any questions for today's lecture? No questions?
Could you go over the slide with the quantitative trait loci mapping again? I didn't quite understand why the marker 1.
Oh.
The one with the
mapping.
QTL mapping.
All right.
Yeah.
Sure.
Yeah,
Let me
I
show
didn't
you
quite understand why. Okay.
This one?
Yeah.
Okay, so before I talk about this slide, let's look at this one. So this one, so for this one, we just compare the two populations, right? So one population is the Yao boys, Yao family. The other one is the other man outside of the Yao boy, right? Yao family. Okay. So if you compare these two distributions, there will be some difference. If we perform a statistical test, for example, the two-sample t-test, to compare this to compare the difference between these two populations, and if the test gives you a p-value is very small, so what does that mean? That means like there's a real difference between Yao family and other families, right? Is that correct? So we are looking for significant results. So we are looking for something that can show us like two significantly different distribution, right? If these two distributions are overlapping like this, you do not see there's a difference, right? But if you see the segregation of these two distributions, then this will give you a strong evidence there's a genetic effects between these two families. Is that right? Two distribution.
Okay.
Yeah. Okay. So now we apply this theorem here. Okay. So for example, this is the sample here. This is a single sample. We cannot do anything for the single sample. However, if we can subdivide this sample into two based on a marker. So here, this is a single marker. So for example, let's use this to represent this marker. So for each individual in this sample, at this location, this marker can take one of two genotypes, either the AA or the AA prime. This is the heterozygous, this is the homozygous, right? Let's assume the red one is the homozygous genotype, and the blue one is the heterozygous genotype. So let me ask you, can we subcategorize, subdivide this sample into two subsamples, subgroups, which carry?
Yes.
Okay, now we have two samples, right? So we can check the distribution of these two subgroups to see if they are different or not. So this will show you difference, but this will give you no difference, right? Because if these two distributions are overlapping, so there's no difference between this one and this one, right? But if you got a distribution like this, so which means there's a big difference between AA and AA prime group. Do you agree?
Yes.
Okay, now, so let's translate this information into the genetic mapping. Oops. So if you got the results like this, which means, so this marker, actually, the genotype of this marker can separate this sample, study sample, into two subgroups with distinct traits, value, right? Human heights. Can we? This marker, based on the genotype of this marker, we can separate the population or the sample into two subgroups with distinct, with significantly different phenotypic values. Look at here, right? And this means this gene or this marker can explain the variability of the phenotype in this population. Does that make sense?
Yes. I was just wondering, does it have anything to do with the ratios between the red markers and the blue markers that makes it significantly like different or not?
Say that again, I'm sorry. Yeah.
Is there anything about the ratios of the red markers to the blue markers that makes it
Right, this is a good question. So here so it could be any ratio. So here I just give you a very simple example. So suppose we have like, so I only give you like 1, 2, 3, 4, 5, 6, six people. So suppose we have 600 people here. So you're gonna have like 600 genomes here, right? So then you're gonna have like 600 dots here. So probably like the ratio of the red and the blue could be like half and half, or could be like 1 to 2, or could be like 2 to 3 or something like that. It doesn't matter. As long as your sample is big enough, you can still test if this marker can separate, can subcategorize your study sample into two subgroups with distinct phenotypic values. So if this is true, that means this gene, this marker, is significantly associated with the variability of the trait.
So like if we were tested on this, would we have to generate the graphs at the bottom by ourselves, or would we be given them and then asked to explain what they mean?
Oh yeah, so the graph, I only give you the graph to help you understand the things behind the test. But when we do the test, we do not have to worry about the graph. We just write the code and let the program to scan the genome based on the data and do the test for each marker, and then we can generate a Manhattan plot to help us to identify the genomic loci that are associated with the traits variability.
Okay. Thank you.
Sure. All right. Any other questions? Yep.
Yes, hello, I had a question. So if we were to consider, in this example that you had just given, we were considering loci 1, but since you mentioned that it's a categorical comparison, could we theoretically use traits 2, 3, and 4 as well in order to conduct the mapping? And then we can't use 5 because all of the loci are the same allele.
Yes, good question. So this, yeah, so right. So this means what? If you got a marker like this is actually not a marker at all, because at this locus, everybody shows the same genotype. So there's no variability in the marker, right? So in reality, if we found this situation for this marker, we're gonna remove this marker from the analysis. So because this marker will not give you any information about the mapping analysis, right? Only when you see the variability in genotypes at a locus, then you can include this marker in the analysis and try to test if this marker is associated with the phenotype or not. Does that make sense? Good question.
Yes, it makes more sense. Thank you.
Yeah, thank you for pointing out this. Yeah, so this I purposely put example here to show you that. So this marker is useless in this study, but maybe when you take another example, when you take another random sample from the population at the same locus, maybe you see the variability in genotypes. It could be, but sometimes, yeah. Because of the random error, sampling error, you take a small sample, and it could be like everyone in this sample show the same genotype at this locus. So in this case, you're going to remove this marker from your study. Yeah, very good. Okay, so if you do not have any questions for today's lecture, let's quickly go over see the practice test. Okay. All right. So I already uploaded this practice test onto the iLearn. So when you have time, yeah, please, yeah, try it, because this will give you some idea the problem styles, the question styles, or what you expect to see next Tuesday on the midterm. All right. So okay. So first, I'm gonna give you the chi-square table for free, so you can check. You can use this table to help you with your calculation, the test. Because as you can see in the first example problem, so we're gonna use this chi-square table to help with your calculation. So this is a classical dihybrid cross, right? Dihybrid, we have two genes here. And I'm gonna give you a table like this. So, all right, anything written in the dark black, so that's the information you can get from me. So I'm gonna show you those informations in the table. But any numbers here in gray, so that's the number which will be hidden from you. So you need to figure out those numbers and put those numbers in the table to earn credits. All right. So basically, I'm gonna so suppose I gave you the number like this. I will ask you to use this number to construct a chi-square test to test the hypothesis. So in this case, we will test like if these two genes are assorted independent a lot, right? All right. This is what we have learned before. So null hypothesis is these two genes are independently assorted, so that the ratio of the four phenotypes, the phenotypic ratio for this four categories should be 9:3:3:1. So first you need to specify the null hypothesis and also the alternative hypothesis, and based on this, you can immediately put these numbers here, right? So you can put like 9 to 3 to 3 to 1 here, or you can say 9 divided by 16, 3 divided by 16, 3 divided by 16, 1 divided by 16. Yeah, that's another way to do this. So it doesn't matter. All right. Now, once you have the expected, so this is the example I showed in class. Once you have this expected value ready, you can calculate the expected value for each category, right? So what we do is like we can add up this observed value together to get the total observation. That's your sample size. And then you multiply your sample size with the fraction here, right? 9 over 16. You can get the expected value for each category. So given this total number, what would you expect to see for this category? 313. For this one, 104, and so on. So once you got this, you can use this formula. So I gave you this formula to calculate the values for each category, and you add up these values to get this, the chi-square statistic. Now you need to know the degree of freedom, because this is a 4-category situation. As we learned before, the degree of freedom equals to the number of categories minus 1. So in this case, it should be 3. And then you just go to the table, just like here, find the p-value, because the p-value is greater than 0.05. So this can help you to make the conclusion. We do not reject null hypothesis, because it's quite reasonable to see the data like this if the null hypothesis is true, right? And so we can say, okay, we accept that these 2 genes are independently sorted during the meiosis. So this is a typical question I could test on you. I hope, yeah, this is reasonable. This is a fair question. I hope you would agree that this will be a fair question. All right, the second question is, so I'm going to give you a situation here. A couple had a son who was normal in phenotype, so you cannot see any abnormal phenotypes. However, so when we check a somatic cell for this male individual, his karyotype shows a little bit different than the normal. So this boy had 1 X chromosome, but 2 Y chromosomes. So this is non-standard sex chromosome setting, right? Okay, now we have 4 questions here. So the first one is, so you need, I ask you to spell out what is the chromosome. So how many chromosomes are there in this individual somatic cells, and describe his karyotype. Karyotype means the makeup of the chromosomes, right? Using the genetic terminologies here. So of course, because other autosomes are normal, the only non-standard situation is the makeup of the sex chromosome. All right, so this one has 46 plus 1, so 47 chromosomes, because he has an extra Y chromosomes here. So, and because of this, so we knew the term of this, right? Because we have an extra, Y extra. So that's a typical trisomy. So you need to put down the trisomy here to get the credit, right? All right, now, so what event in the meiosis will result in this unusual karyotype? Yes, we have learned this because the reason why one reason for the trisomy is because of the non-disjunction. So the non-disjunction resulted in the gametes with 1 extra X chromosome, right? And so the part C, I will ask you, okay, so yeah, based on the information, can you determine in which parent and in what meiotic division stage could this event have occurred, right? So that's easy because this can only occur in the so this non-disjunction can only occur in the in father at the second stage, second meiotic division, cell division, right? So and the third part is the major part because I need to I need you to show you fully understand the process. So you have to draw this diagram to show me you completely understand what is going on behind this. Okay, so this is the father's karyotype. So one is X and one is Y. So this is X chromosome, this is Y chromosome. So in the first division, so X and Y chromosome are gonna separate, ending up into two different cells, right? But at the second, the meiosis 2, second cell division in meiosis. Okay, this is X chromosome, yeah, just normally separated into two gametes. However, if the non-disjunction occurs at this cell, so these two Y chromosomes, the sister chromatids, do not separate. They come together into the same gametes. So these gametes will carry two Y chromosomes, and this is the chromosome. This is the gametes from the father. Join the mom's egg to form a non-standard zygote with two Y chromosomes. Okay, any questions here? So as you can see, the problem like step by step. So the question will lead you to find the final answer in a logical way. All right, this is another question. This is another field that I can ask you because we learned the human genetics and we learned how we can study a pedigree to help us to calculate the probability of any matings. All right. So I just gave you the exactly the same problem that I have given you in one of the quiz. So this is a typical problem. Of course, I will give you a different situation. So, but if you understand what is going on here, and if you can successfully get the right answer, then you should not be able. You should not worry about the midterm next Tuesday. So this is. So again, so let me briefly talk about this. All right. So here, so we're talking about so this male and this female, they want to marry, and they just want to figure out, so what is the chance that their first kid will have the disease? Because this man has a sister who is affected by the disease, and this woman has a brother also affected by the disease. This is a autosomal recessive disease. So those are the two individuals affected by these two families so far. Their parents, both of their parents are normal in phenotypes. They should be carrier, right? Because the heterozygous, the carrier can give a affected kids in both family. Now we want to calculate the probability that these two normal parents will give a affected kid because of their phenotypes. They are normal, so we need to understand why each of these parents will have like 2/3 of the chance to be a heterozygous. So you need to understand this. Just draw the classical monohybrid cross Punnett square to help you to get this probability values, and finally, yeah, you can get this answer. So, all right, and we have four questions in the midterm. So this is how the real test will look like. And in the last part, I'm gonna ask you to briefly define some definitions, some concepts. For example, I will ask you to define a gene or an allele for me. So the best way to prepare for this problem is like go over the slides, the lecture slides, and find the important concepts that I have covered, and make sure you understand those, and then you will be doing great. Yeah, in this part. All right. I think the time is up. Any questions?
Hi, Professor. I was wondering just how like similar the practice midterm is to our midterm on Tuesday.
In terms of the format, very similar. So I'm going to give you exactly four questions in the same style, but yeah, definitely. I'm going to give you different problems, different situations, different cases. Yeah.
Okay. And will there be like different concepts that you'll be testing other than the ones you showed on this midterm?
Oh, you mean this?
Like on the I don't know, for example, like would you maybe like throw in like something that we went over today?
Yeah, so today, yeah, of course. So the first midterm will cover what we have learned so far from chapter 1, from lecture 1 to lecture 6. Yes. So pay attention to, so I have already uploaded the video online on YouTube. So if you have time, so you can briefly go over each of those, because sometimes when I say this concept is important, so yeah, I meant that. So
Okay.
probably I'm going to throw that concepts in the exam, so
Okay.
to test you.
That's good. Thank you.
Sure. Any other questions? And tomorrow I'm going to have my office hours in the afternoon. So in case you have questions, you can come to my office hours. We can discuss, and I can help answer your questions. All right. Thank you. Have a good day. Bye-bye.
Hello, Dr. Jaya. If it's not too much to ask, would you please be able to focus on question 3, solution?
Question 3. Yeah, sure. Let me share the question 3, right? 2, 3. Okay, this one?
Yes, thank you.
Oh, sure. So okay, so for this one, we are talking about a autosomal recessive disease. As you can see, so we are interested in this family, right? So this man and this lady, they are normal. Okay, the problem is, so this man has a sister who is affected by disease. Then we can immediately put down the genotype for his sister, right? A little bit. This is homozygous recessive. All right, the same thing for this lady's brother. So because this brother, her brother is affected by the disease, so we can put down the genotype little bit here. All right. Okay. Now, since so we can extend this problem to here. So look at this mating. So the man's parents, they have a affected individual. What does that mean? That means like both of them should be heterozygous. Otherwise, it's impossible for this family to have an affected kid, right? Do you agree?
Yes, and because the parents of the F2 progeny, because they are heterozygous, it means that we cannot consider either parents having a homozygous recessive characteristic. Therefore,
that's true.
That's why they would only have a 2/3 chance of passing on the characteristic rather than.
That's exactly like that. Yes. So because these two, yeah, because they are normal. So there are two ways to be normal, right? Either they are homozygous big P, and another way is like they are also heterozygous, right? But if we want to calculate the probability that kid will be affected by the disease, then we need to calculate the probability that this parent and this parent are heterozygous from these matings, right?
Yes.
So this is how we understand this problem and get this calculation here.
All right, thank you for the information.
Sure, yeah, sure. Any other questions? So I can stay for another 5 minutes if you have questions.
Professor, I can't
Yep.
find the solution manual for the practice midterm you just uploaded. Can you please upload that too?
Yes, I just finished this this morning, so yeah, I will upload this to Island as well.
Thank
Sure.
you so much. And then I just wanted to double check. I saw in the syllabus that the Fridays before midterms, we have a genetics clinic, so that it will be tomorrow from 4 o'clock to 4:50.
Yes, so usually I will extend my office hour for another hours for another hour if needed. So sometimes like I don't see students coming, so I'm gonna just have 1 hour's office hour. But tomorrow I believe many students will have questions for me, so I'm gonna extend my office hours to 5 p.m.
Okay, thank you so much.
Sure. Okay. Bye bye. Any other questions? Okay. Have a good one.